Just thank everyone for being here today. I want to take a moment again just to thank Sudran and her team. It takes, some of you know this, but it takes an incredible amount of effort to make a day like today happen. And there's a huge amount of work going on behind the scenes, Hallie, Ella, and team. So can we just give a quick round of applause for them? Thanks so much. And so I'm the chair of the board of Aim High. I've been doing that for 5 years. You just saw, I think, the most exciting part of that program throughout the year is to recognize the exciting women leading these companies. But what you may not know is throughout the year, there's a huge amount of mentorship. And this was really created out of NFCR's program to put a sharper focus on entrepreneurs and supporting them. We do today's event, and throughout the year, there's a number of people, Sudran, I can't name everyone, but Sudran, Raju, Pam, spending a ton of time with entrepreneurs and helping them to sort of make their way through the ecosystem as they support their companies. So I want to shine a spotlight on that. Today, so we've heard a lot of exciting science today. Where are the frontiers of cancer treatment being pushed to? We're going to turn for 2 sessions this afternoon to hear about sort of more about the business of cancer treatment. We have executive leadership from 5 companies to present. Several of them are companies in which Aim High has invested. I run an organization called Blackbird Laboratories as my day job. We've also invested in several of these and really have a synergistic function with Aim High here in the Maryland ecosystem. I was going to say a few words about Blackbird. I'm going to save us some time, but if you're interested, come and talk to us during the breaks. My team is here as well. So with that, I'm going to jump right into introducing our first presenters. We actually have a 2-for-1 Adventris Pharmaceuticals, Jen Herbach and Mark Yarchoan, who are co-founders along with Liz Jaffe of Adventris. Very briefly, Jen is a Harvard-educated MBA. Prior to Adventris, she was leading corporate development for Amunix, which was acquired by Sanofi for $1 billion in 2021. Mark did his medical training at UPenn and Johns Hopkins, and in addition to his role at Adventris, is a physician and faculty member at Johns Hopkins. So we'll welcome him to the stage.
All right. Good afternoon, everyone. So thank you to the organizers. We're here on behalf of Adventris, and Jen Lesser is our CEO, so I'll let her introduce herself.
So thanks so much. It's great to be here. I'm Jen Herbach. I'm one of the co-founders and the CEO of Adventris. And
I'm Mark Yarchoan. I'm a physician scientist at Johns Hopkins and a co-founder with Jen of this company.
So thanks so much. It's so good to be here, and we're so excited to introduce you to Adventris. And really simply, we make cancer vaccines. Okay, which is, thanks. So before we dig in, I'd like to highlight kind of what we think are some of the 5 key differentiating factors of Adventris. So first, we have built a team of world-leading cancer vaccine experts, one of whom is nicely standing next to me today, and I'll introduce you to the rest of our team shortly. But when we think about what's so unique about us and what differentiates us and our special sauce, it's ultimately our cancer vaccine platform that enables us to solve one of the biggest challenges in the cancer vaccine space, which is the targeting of poorly immunogenic oncogenes. So given this, not surprisingly, the first target that we're going after is KRAS, the most common oncogene, and this accounts for about 30% of all cancers. So we're starting our clinical development plan in adjuvant lung, colon, and pancreatic cancer. And most excitingly, we actually have preclinical as well as clinical data that supports our approach. So as I mentioned, we've built a team of world-leading cancer vaccine experts in addition to Mark. This company is really built off of decades of Dr. Liz Jaffe's work and is a spin out from her lab. She needs very little introduction in this group, the co-chair of Biden's Moonshot, the past president of AACR, and she's been an incredible scientific leader of this company. In addition, we've raised a $12 million seed round from a strong syndicate of investors, and very thankful that our investor Matt and Blackbird invited us to speak with you guys today. So in addition to our scientific leadership team, we've built a world-class advisory board, including leaders like Dr. Patrick Hu and Dr. Kathy Wu, who's been a world leader in the neoantigen vaccine space. Now I'll turn it over to Mark to kind of dig in more on the science.
So, everyone here in this room is probably familiar with vaccines, but mostly in the sort of virus realm. And when we think about cancer vaccines, people may be familiar with like the HPV vaccine or the hepatitis B vaccine, which prevent viruses that cause cancer. But I think for the first time, there's real enthusiasm in our field that we may soon have a number of effective vaccines that actually directly treat or prevent cancer. And at the end of the day, what a vaccine is it's something that educates the immune system to recognize a target. And increasingly, I think there's optimism in our field that that may be possible for direct targeting of cancer antigens. And for those of you who have followed the space, there's a lot of enthusiasm in part because Moderna, which is now of COVID fame, has turned its attention to cancer vaccines and has a positive randomized trial and is moving quickly towards potentially approval for at least one cancer. Our approach is a little bit different. A lot of the enthusiasm right now is around personalized vaccines, and we certainly think that this is an approach that carries some water, but we really believe that the best antigens to target with a vaccine are true cancer drivers. These are the things that actually make a cancer grow. And that's because if you get the immune system to recognize the driver of a cancer, because the cancer is addicted to this thing, it can't get rid of it. And so you can get a deeper, stronger response than is possible by targeting whatever you find on the cancer cells other companies are trying to do. As a practicing oncologist, my dream is to put myself out of business. And when you think about how are we going to do that as a field, and I know prevention has been a major theme of the day, when you think about designing a vaccine, vaccines work best for prevention, not for treatment. But to make a personalized vaccine, you have to first have a cancer. You can't use a personalized vaccine for a person who hasn't developed cancer yet. If we could make a vaccine that targets the drivers of cancer, we could use it not only to treat cancer or treat very early cancers, but in the very long run, we could use it to actually prevent cancers. And our first target, KRAS, is the driver of about 25 to 30% of lethal cancers in the adult population. So if we could just make the body see this one thing, we could eliminate 30% of all cancer deaths. So seems pretty easy, right? Make a vaccine to target KRAS. Why isn't everybody doing this? Why has this not been successful? And the challenge is, at the end of the day, our immune systems are pretty good at seeing things that are abnormal. The reason we get cancer is that our body fails to see these drivers, right? These are fairly poorly immunogenic antigens. So how do we deal with this? So we believe that we have a technological solution that allows us to target these very challenging things like cancer drivers, and that's our Adventris antigen engineering technology that I'll introduce you to in a moment. So how does this work? Again, there are some immunologists in the room, some non-immunologists. I'll try to simplify something. So all the way there on the left, you have a standard peptide in a standard cancer vaccine. That is a KRAS G12D, let's say. So right in the middle, that red ball is the abnormal thing that the cancer has that we want the body to see. So every other vaccine platform that we're aware of, you take the thing that's in the cancer and you give it as a vaccine. At the end of the day, our thesis is if the body hasn't seen this already, it's unlikely to respond well to a vaccine. So what do? We take the thing that we want the body to see and we actually modify it. And we modify it in a way that generates a cross-reactive response with the parental antigen. So this is the principle of molecular mimicry. Some of you may be aware of this. You get an infection, and then after the infection, your body starts attacking itself. That's how rheumatic fever happens. It's a very powerful immune response. We try to do that intentionally, but instead of attacking ourselves, we want to attack the cancer. So why do we believe in this? Our company is really a spin-off from Johns Hopkins, and our academic group has been developing vaccines for some time. And consistently what we've seen in these clinical trials of our first generation vaccines, this does not use our antigen engineering approach, is that a subset of patients mount responses to these vaccines. The patients who mount a response to these vaccines do better than the patients who don't mount a response to these vaccines. And that's something we've seen in a trial in pancreas cancer that should be published soon. It's also been published by other groups, such as the recent BioNTech trial in pancreas cancer. If you mount a response to a vaccine, it's meaningful. So our thesis is we need to get more people to that threshold of clinical benefit to really make a difference in the cancer world. So how did we come up with this idea? This was really a mistake. So there's no better way to learn from your mistakes in science. So this is work from a PhD student in Liz Jaffe's lab. Her project was to try to see what checkpoint inhibitor to combine with a vaccine. So she made a personalized vaccine, very similar to what Moderna or BioNTech are making. She ordered a bunch of peptides to encode all the antigens in the cancer, and she gave the vaccine, and all the mice started clearing their tumors. It was very exciting. And then she realized a couple months into her project that she had made a big mistake, and 2 of the peptides, she had ordered the wrong sequence. So these were incorrect peptides. So, PhD students have a lot of time. She came forward, she said she made a mistake, she ordered the correct peptides, and suddenly the vaccine stopped working. So the wrong vaccine was clearing the mice, the correct vaccine had no efficacy. What was going on? It turned out that the incorrect vaccine was much more potent, was much more recognized by the immune system, and generated a cross-reactive response against the correct sequence. Just a total accident. So we figured if this is so powerful, even accidentally, we can do this intentionally. So we think we're pretty unique. There are a lot of companies developing neoantigen vaccines. This is suddenly a very hot topic in our cancer world. I think the personalized vaccines clearly are very exciting, but probably are going to work only for people with established cancers, and likely only in tumors that have a lot of neoantigens, a lot of targets. We really think that our approach is quite different. We're the only ones modifying the antigens that we know of, and we think that this allows us to go into cancers that don't have a lot of antigens, these cancers that only have these drivers and not a whole lot else. And then in the very long run, our grand vision, again, is not only to treat cancer, but eventually to prevent it. And let me tell you, after being a practicing oncologist for a number of years, there's nothing that gets me more excited than putting an end to cancer. So I'll turn it over to Jen.
So thanks so much, Mark. So we'll just kind of walk through where we're at as a company today. So in terms of our antigen engineering platform, we have validated it to date, and we've showed both enhanced immunogenicity of our engineered antigens in vivo, and also showed improved survival and tumor reduction as well in vivo. We're currently working on our KRAS vaccine right now, and we've showed enhanced immunogenicity of our KRAS antigens in vitro. We're going to be targeting candidate selection of our development candidate in mid-2025, and then actually dosing our first patient in 2026. And I know, I just wanted to finish up with kind of one last topic. As I know Mark mentioned, I think we're a little bit unique among companies in the cancer space in that while we're initially focused on treatment, I think why we're all here and why we're doing this company is our ultimate long-term goal is really to be able to move these vaccines from the treatment stage into the prevention stage. And to be able to be a part of that and basically put Mark out of a job. So we're obviously focused first on our KRAS vaccine for treatment, and then with the goal of moving that into the prevention side, and then eventually to be able to actually target the most common oncogenes, put that on one vaccine, and move that into the prevention space. So we've set our goals just a little bit high. But yeah, thank you so much for we're so happy to be here today. We'll be around all day and at dinner, so if you have questions, please come find us.
Great, thanks a lot. And if we do have time at the end of this session, we'll bring the presenters back up for some brief questions. But in case we don't, as Jen mentioned, try to find these folks during the breaks in the reception and talk to them about their companies. Next, I want to welcome to the stage Dr. Samir Khleif from Georgiamune. He's the founder and CEO. Among other things, Samir was a researcher at NCI for over 20 years and among other accomplishments, helped to create the famed NCI 60 tumor cell bank. He's held several faculty appointments over the years and is now, in addition to his role as founder and CEO of Georgiamune, is a faculty member at Georgetown. And I've asked Samir in particular to tell us a little bit about his journey, because I think it's really emblematic of sort of the bridge between the two worlds that we're hearing about today, cutting edge academic science and really translating that into biotech companies that are putting drugs into the clinic. So thank you, Samir.
Thank you. So first, I'm honored to be here, especially March 40 looking over my back. So I look behind and I see her, and I think that's really an honor. But it's great to be here. It's great to be part of all of you. So Matt asked me to talk a little bit about the career. I started my career at NCI, and where I was lucky to be there since it was a cradle for immunotherapy development. Some of the attendees were there today, and I had the great honor of being part of them. I was asked to start the first cancer vaccine program at NCI. At that time, as you heard from Jenny and Mark before, it used to go oncogene cancer vaccines. Now, affectionately, we call it neoantigen cancer vaccines. And we started the first clinical trials in cancer vaccine programs for mutant RAS, mutant P53, VHL in the 1990s. It was a major learning experience. When I was at NCI, I was also asked to go to Jordan to build a cancer center in Jordan, which I did, and it helped me a lot. I think more confirm some expertise, or at least learn how to develop institutions and how to put together institutions out of nothing towards building it into to be an effective institution, which helped me a lot in my career by far. The other thing that I was very lucky to do at NCI is I was detailed, and Anna Barker is here, she and I worked together on this to the FDA halftime to lead the critical path, which I learned tremendously about drug development and about how to think of drug development from not only from scientific perspective, but from regulatory perspective. After NCI, I was recruited to lead the cancer center for University System of Georgia. And I worked there for almost 6 years until basically I looked one day and found that we have a lot of IPs that we've developed from my lab. And I had one of two choices. Either I'm going to leave it to the institutions to add it to the cemetery of IPs that majority of our institutions, unfortunately, is good at, or to take the bull by the horn and form a company and move it forward. And henceforth, this is what I decided to do. And by the way, this is where Georgiamune come from the Georgia Cancer Center, where I was a director there. So what is Georgiamune? So Georgiamune is a science and discovery company that we formed it based on IP that first we licensed out of my work at Georgia, and we added a lot of IP to it during the development of the company. It is science and discovery means it is a company that tried to discover completely new science, and therefore we are able to develop first-in-class drugs because of that new science in immunology. So it's an immunotherapy company, and it's an immunotherapy company for cancer and autoimmune diseases. So we have two state-of-the-art biologic platforms, and that led both of them to the development of first-in-class drugs. The intent is, again, to develop those first-in-class drugs for unmet need in oncology, for areas, or autoimmune disease, for areas where we know that there are not too many things to be able to offer our patients. So what we did is we have, based on all the discoveries, we have developed a very robust intellectual property, more than 240 patents in 24 different families. And we were able to raise $100 million so far as part of Series A for the company. Currently, we have 3 drugs that are already in clinical trials that we were able to drive into clinical trial in the past 12 months. So the company came out from stealth almost a year ago, and I think we were pretty lucky to be able to get pretty prestigious awards because of the platform, because of the path of development, and because of the pipeline that we've developed so far. So these are the two platforms. One of them is novel functional T cell receptor epitope discovery. The other one is T cell molecular discovery of master switches of immune cells. We do integrate everything we've been doing from a drug development perspective or from identification of targets with a very broad AI programs or AI platforms. So because of those kind of discoveries and their fundamental immunologic discoveries that we had, eventually, when we identify those targets, we can literally take them one way or the other. They could be helping in activating the T cell, because the T cell is underactivated in majority of our cancer patients, which means to try to program the cell to become either more active or a different type of T cell. Or, since the other side of the coin is autoimmune disease, where the cell is overactivated, that fundamental biologic discovery can also be toned down by leading to the decrease in overactivated T cell, or reprogramming T cells to become helpful in autoimmune diseases. So based on that, the company now has 11 drugs which we developed in the past 4 years, 4+ years, maybe 5 years in development. 3 of them are already in clinical trial, 2 for cancer and 1 for autoimmune diseases. And 2 drugs are already in declared as drug candidates, so we're taking them into IND development, and the rest are in lead optimization. And all were developed based on fundamentally new science that we have discovered at Georgiamune. So again, Matt asked me to talk about the team and how we constructed the team. So we have really a very strong team of leaders, including Rami Ibrahim, who's a very well-known chief medical officer. He used to be at AstraZeneca and BMS. Jacques Bonchereau, who's one of the lead immunologists in the past 30 years. He had many contributions to immunology, and Mika Mukherjee, who is an expert in both small molecules and large molecules. We have a great board of directors. Some of them you might know, including Amy Abernathy and Sean Parker. And we have a fantastic scientific advisory board that really encompasses the knowledge of clinical and basic science development in the field, but also including drug development and thinking about we develop drugs, including Mace Rothenberg. Some of the people that worked at NCI know Mace very well, and Elliot Siegel, who used to be the executive vice president at BMS. Because we are clinical. 2 clinical paths, including oncology and autoimmune diseases. We have put together experts in both that are working with us very closely on not only helping and thinking of how we design clinical trials, how we determine indications, but also we engage them with our science from early on to think how we can be, or what we need to be developing every product toward clinical development from the get-go, and think about what kind of preclinical studies, translational studies need to be done early on to determine where we go forward with this. Now, one of the major successes that I believe that helped us to lead to the development of 11 assets so far, or 11 drugs so far, is really getting the experts in the world of how to develop drugs. So our small molecule experts that work with us literally on a daily basis are who's who in the development of small molecules. Some of them actually worked at Merck in the 1990s and 2000 to develop small molecules at Merck. Our large molecules are the same. TJ is worked 10 years with David Baker to develop large molecules and understand protein chemistry and protein structure. So I believe this is very important when we develop any sort of an endeavor to develop drugs, that those experts are not only crucial but mandatory. These are our investors, including, of course, Blackbird, where Matt comes from, and others that are big part of so far what we've been able to do. So just going fast through the first or the ones that are in clinic so far. The first one is we call GIM-122. It's a monoclonal antibody, and because of the novel biology that we discovered, it has a dual functionality within T cell. Why this is important? It is important because as we know, and I'm going to skip through this very fast, but most importantly is 85% of patients still do not respond to checkpoint inhibitor. And because when we think about checkpoint inhibitor, we think that in general, the people that respond to checkpoint inhibitors are those that have activated T cell in the first place, which means patients with hot tumors or tumor mutational burdens, i.e. the tumor is properly primed, properly activated. So when it goes, when this go to the, when the T cell go to the microenvironment, it gets suppressed by the checkpoint. And then when we block it, we can regain back the activation status. The problem, though, is majority of our patients do not have activated T cells, whether cold tumors, low tumor mutational burdens, and others. So when it goes to the tumor, get blocked, get inhibited, it's not going to get inhibited because it's already inhibited. And when we release that inhibition, nothing's going to happen. The question is, how can we overcome this by a ability to reactivate that T cell and make it functional within that tumor microenvironment? And this is what the biologic discovery of 122 lead to. So now it is in clinical trial. We are conducting our phase 1 already in multiple centers around the country. And the plan is as soon as we finish the phase 1 escalation, which should be very soon, we will be starting this in different indications. The second program that's already in clinic is targeting Tregs. So we were able to discover a molecule that regulates Treg very intimately. And based on that, we were able to develop drugs that selectively inhibit Tregs and selectively activate Tregs. So the first drug is that I'll show you is a cancer drug, of course, because in cancer we need to inhibit Tregs, since Tregs is overactivated, and it leads to one of the mechanisms of suppression of the anti-tumor immune response. And I'll skip that. And the drug that we have now is an oral medication. It's called 531. It's a selective Treg inhibitor, and it inhibits both the numbers of Tregs and the activity of Tregs, which is very, very highly selective, and more importantly, very unique within the market, since there's none like this. So the current market, majority of the Treg inhibitors or targeting Tregs target the phenotype of Treg, which lead most of the cases into also the suppression or the depletion of the activated or the effector part of the T cell repertoire, including CD8 and CD4. And this is why 531 is unique. And if you look at this, when we give it to mice, we can decrease the Tregs in a dose-dependent manner, but also the functionality, and it is very specific. It does not affect neither CD4 nor CD8. So, the interesting part, it works as a single agent because that actually reflects into the tumor microenvironment exactly as I showed you on the Treg and CD4 and CD8, and it leads to the activation of CD8. And if you compare it head-to-head with checkpoint inhibitor in multiple tumor models, usually it shows a much higher potency in tumor models. So based on that, we are currently in clinic, and that's our second clinic for the past clinical trial. And we are in dose escalation. As soon as that will be done, we will be going into monotherapy in 2 different indications and in a rescue approach for PD-1, because as you know now, also one of the mechanisms of inhibition of resistance to PD-1 is the number of Tregs within the microenvironment and even in the periphery, as a lot of data came out in the past year. The third program that I'll share with you, which is last, is our first in the market selective Treg activator. Because of the discovery, we were able to develop small molecules that activate Tregs. And of course, where we need Treg activated in autoimmune diseases, because majority of autoimmune diseases, the Treg is dysfunctional, it's not working well. So we need to activate that Treg and increase the numbers of Tregs. Again, there are many patients that needs and have unmet needs for Treg activator. The drug that we have is called now that we've developed is 407. It is an oral drug. It's given daily, and it activates Tregs that are dysfunctional. In addition to that, it increases the number of Tregs because it helps CD4 to convert to Tregs. So with that, this is the first in the market, since there are no drugs in the market that do both of those at the same time, dependent on the mechanism of Treg regulation. So we took it into clinical trial. I'll just show you. So this is in mice, and you can see that there is exactly the opposite of 531, where you have increase in T regulatory cell, and it is a specific increase in Tregs, and it does not affect CD4 and CD8, and that reflects into the end organ, which is affected by the autoimmune disease. So just to show you an example of autoimmune disease that we treated with 407 in the preclinical settings, and we've treated now most of the autoimmune diseases. This is diabetes type 1, which is the NOD mice model, and we've treated more than one specific for diabetes. But you can see this is the islet cells in the pancreas of naive mice where the pancreas is functioning very well. This is the diabetic mice where the islet cells are completely infiltrated with effector T cells, and it's completely destroyed, leading to increasing glucose within almost 30 weeks to almost 600. And all mice within 25 weeks become fully diabetic. All those mouse models we treated therapeutically, not preventively, so that's really very important. Now, if we treat with 407, we can find Tregs are increasing, periphery and within the tumor, within the end organ microenvironment. And you could see that the islet cells are coming almost back to normal. They're secreting insulin, and the mouse are completely diabetes-free within the 25 weeks to 30 weeks, which is the same period of time. So currently we're conducting this in healthy volunteers because we have to do SAD and MAD in autoimmune diseases, which is something that I've learned since I'm an oncologist. I had no idea what and how to approach autoimmune diseases, but because of our experts and our KOLs, we are currently conducting this trial, and as soon as that happens, we will go next to specific indications that we are determining what they are as we speak. So again, this is where we are. We are taking the rest of those into IND very soon, and hopefully we will develop them into multiple different indications. Thank you very much.
Great. Well, thanks very much. Really excited to welcome to the stage now Dr. Vipin Suri, the Chief Scientific Officer of Clasp Therapeutics. And it's another great connection amongst our community here. We heard a whirlwind tour from Drew Pardall this morning about the science behind the mutation-associated neoantigens. And we heard a presentation last year from some of Dr. Suri's colleagues. And so we'll hear now the great progress that Clasp Therapeutics has made.
Thank you, Matt, and thanks for inviting us to share our story at this wonderful conference. It's been great. I've certainly learned a lot, so thank you. So Clasp is about a 3-year-old biotech. We have a presence in Rockville, Maryland. We certainly have our founders and collaborators at Johns Hopkins, and we're based in Cambridge, Massachusetts. We're developing T-cell engagers for solid tumors. So we're big fans of immunotherapy for all the reasons that Drew and others mentioned. We believe this is a great and proven approach for deep and durable responses, particularly in solid tumors. The problem we're interested in solving is how do you bring exquisite specificity to immunotherapies so you direct them only to eliminate cancer cells and spare normal cells. Okay, great. Mark and Jenny did a great job explaining driver mutations and neoantigens, so thank you for that. That makes this slide a lot easier. But one of the features of tumors is you frequently see mutations in oncogenes such as p53 and KRAS, which are frequent in a given tumor type and also across many different tumors. So p53, for example, is mutated in about half of colorectal cancers or pancreatic cancer or non-small cell lung cancer. And just to be clear here, this is not a single mutation in p53, this is all the mutations in p53. KRAS similarly is mutated in about 80% of pancreatic cancer. And the features of these driver mutations are they're exclusive to cancer cells. So they're not present in normal cells, they're only in the cancer cells. They are a requirement for the cancer. So the cancer cannot avoid this mutation. The cancer needs it to proliferate, right? And they're present across the tumor sample. So they're present in all cancer cells. As Mark explained, the presence of these mutations in a given cell can be recognized by presence on the cell surface of these neoantigens, peptide HLA complexes on the surface of these cells, right? So these cells are marked by these peptide HLA complexes, the cells that have these mutations. If there was a way to recognize with an antibody-like biologic cells that have these markings on the surface and then direct the immune system to eliminate those cells, then you have an immunotherapy that's very precise and specific for cancer cells, and it spares normal cells. So the approach that we've taken is to develop these antibody-like biologics, which we call flares. FLAIR stands for Precise HLA Redirecting Engagers. And these flares are antibody-like biologics with 4 key features. One, they have these anti-peptide HLA moieties. So these are variable domain sequences that can recognize peptide HLA complexes with high specificity. They have a CD3 moiety that engages immune cells. So when this flare sees the cancer cell, it will engage the T cell to launch a response. They have a IgG4 moiety, which increases the half-life. So these need to be dosed every 2 weeks or every 3 weeks. And the fourth feature is they manufacture just like antibodies. They're manufactured at high yields, straightforward purification. And I say this not just theoretically, it's because our first product is headed to clinic in a few weeks. We just finished making GMP batch, and it looks exactly like you would make an antibody. So we optimize these flares on 3 key metrics. One is potency, which means that when they see cancer cells, they should be able to launch a very potent T cell response. The second one is sensitivity, which means that they should be able to recognize a cancer cell, whether it has 2 peptide HLA on the surface or 2,000. And the third one is specificity. So they need to be exclusively specific for the neoantigen, not just versus wild type, but versus every other peptide from the human proteome that can be presented by that HLA. And the way we are able to optimize for these 3 features is 2 key technical advancements. One is the quality of these peptide HLA binders. So as Drew mentioned this morning, Bert showed it could be done. We've actually industrialized it. So we've shown it can be done for pretty much any peptide HLA antigen, and you can pick these really good binders. The second is we spend a lot of time optimizing the geometry of how we arrange the peptide HLA with the CD3 moiety to get the best functional response we can. And where we landed, and we've actually done structural characterization and modeling, is that the optimal arrangement looks very similar to how a TCR recognizes its target cell. Okay, and then, so now that we have this platform, we've started to apply it to therapeutic situations. Our first product is a p53 flare. p53 is the most common driver mutation across most tumors. This product recognizes p53 R175H mutation in the context of HLA-A02. So p53 is the most common oncogene. R175H is the most common mutation in P53, and HLA-A02 is the most common HLA type in Caucasian populations. The reason we like this product is that there's a high unmet medical need in, for example, colorectal cancer. And P53 is especially associated with microsatellite stable colorectal cancer, where the checkpoint inhibitors don't work as well as pancreatic and other cancers. We also find this is a compelling commercial opportunity. So, one of the questions that comes up is, if you're being precise, then you can only help a very small number of patients. We've actually done the analysis here, and the patient numbers here present a significant opportunity. And then on the right here is some of the preclinical data. This is a mouse model where we implanted into a mouse that has a humanized immune system a tumor that expresses P53R175H in the context of HLA-A02. We let the tumor grow to a certain size, and then we give it our drug. And as you can see, the tumor melts away. The three lines you see, one is the control line, where it says 1025 1x. We gave a single dose to that mouse, single dose of the drug, and you can see there was a pretty good efficacy. The blue circles is when we gave 3 doses of the product to the mice about a week apart, and then that's where you see regression. The tumors are actually barely noticeable. The second application is KRAS, again, for all the reasons that Mark and Jenny mentioned. KRAS is a high unmet medical need. Pancreatic cancer especially, it's come up a few times today. About 80% of pancreatic cancer has mutations in KRAS. KRAS, generally, we are pretty excited about the space from the development from other approaches as well. Small molecules for KRAS are now on the market for G12C. G12C is one of the more rare mutations. G12V and G12D are the more common mutations. Our product is directed towards G12V. What's shown on the right here is some data from our preclinical studies. And the reason I wanted to highlight this was one is what's shown here is we take a cell line that expresses KRAS G12V in the context of HLA-A03, which is what this product is designed to do. And then we co-culture that cell line with T cells. Then we add our product, and then we look for activation of T cells. So when you see the signal go up, that means the T cells are getting activated as they see the cancer cells in context of the product. There's 4 different T cell engagers shown here. The blue line you see at the bottom, the blue circles, that was our screening hit. And our affinity maturation engine now can go from that hit to the black line, which is our candidate, within a few weeks. So the combination of industrializing the binder discovery and then setting up a platform that can quickly do affinity maturation sets us up well to tackle many such neoantigens. And that's what we've done. We've actually expanded our pipeline quite a bit because we're getting more and more comfortable in designing these sort of flares. And the flare architecture is entirely modular, right? So the only thing you change for these products is the peptide HLA binder. So once we've cracked the code on the peptide HLA binder discovery, rest of it becomes plug and play. The last line here is a viral antigen. So far I've talked about driver mutation neoantigens, which is a very significant opportunity. We also believe we can make a difference and offer some good therapeutics in the situation of virally driven tumors. We have a wonderful team. We're about 40 people, split about evenly across Rockville and Cambridge. What we're really blessed with is a fantastic group of founders and collaborators. Fortunately for us, Drew and Bert fought the battle for a decade or many years, developing the techniques so you can find those peptide HLA binders, and then we have the good fortune on building on that story and industrializing it to bring therapeutics to patients, hopefully over the next few years. We're also very grateful to our investors. The list is there. Blackbird especially has been very kind to us from the beginning and very supportive, which we're very grateful for. And just to summarize, we have this proprietary flare platform, which is optimized for potency and half-life. we're applying that platform to important areas of unmet medical need in KRAS and P53 and many others. We are, again, fortunate to work with the Johns Hopkins team as well as a great group of investors.
Great, thank you so much, Vipin. And I apologize, in my haste, I failed to introduce Vipin's background, but as you saw, he's a very capable chief scientific officer, having spent time in both biotech and big pharma prior to that. So thank you so much for that. Next, I'd like to welcome to the stage Dr. Lily Zhou from Degron Therapeutics. She's the co-founder and CEO. And this is another exciting ongoing story within the AIM-High and NFCR world. We heard from Lily last year about Degron's progress and recognized the great work she was doing. And we're excited to hear about the progress that's happened in the last 12 months. Lily has served in many roles in big pharma and in biotech prior to founding Degron, and has graduate degrees from both Cornell and MIT in science and business. Thanks, Lily.
Thank you, Matt, for the introduction, and I'm glad to be back. We were one of the winners of the AIM-High Women Entrepreneur Competition last year, and I can't believe it's been a year already, but really glad to be back to this really exciting conference. So Degron is a drug discovery and development company focused on the in the molecular glue degrader space. Very quickly, I'm glad one of the winners this year is also a molecular glue company. So very quickly, what is a molecular glue? Molecular glue degrades the target of interest. So if you can, there's no pointer, if you can see from that, traditional inhibitors inhibit a function of a protein, the function of the protein by occupancy-based inhibition. But molecular glue and another technology called PROTAC, they bring together two things. They bring together the E3 ligase of the proteasome system, the protein degradation complex, together with the target of interest. So by bringing together these two proteins, it induces the E3 ligase to act ubiquitin to the target protein. And then once tagged with ubiquitin, proteasome will recognize the target protein as garbage and will degrade it. So there are mainly two technologies, PROTAC, which is a linker, linking one end something attaching to the E3 ligase, the other end attaching to the target. And molecular glue is a pure small molecule. It acts as a glue to bring two proteins together and induce this protein-protein interaction. So why do we do molecular glues? It solves the problem of undruggable targets, because most protein disease targets they don't necessarily have this pocket and the function to be inhibited. So traditional inhibitors, they have to inhibit a kinase or something with function. But degraders can get rid of the protein. And for molecular glue, it doesn't need a pocket to bind either. So this glue can target those transcription factors, scaffolding proteins, traditionally undruggable targets. So it expands the target space for diseases. And comparing to PROTAC, another advantage of glue is a pure small molecule. The drug-like properties are usually better, similar to small molecule. Doesn't have the challenges of the PROTACs. Degron has been working in this field, and we developed a drug discovery platform that help us discover novel degraders for new targets. As I described, it's a ternary structure formation because a small molecule have to bring two proteins together. So this drug discovery currently cannot be rationally designed. We have to use a library and a screening platform to find novel degraders. We've built a very focused glue library which is structurally differentiated from most companies' molecular glue compounds that are mostly imide-based, has the imide-based core, similar to the lenalidomide core structure. Our company was initially founded when we first discovered a different core, has a different core structure that can degrade a protein called V1. Then built on that, we expanded the chemical space and built diversity in that molecular glue compound library. So now we have 10,000 compound library, which are all glue degraders. We don't know what they degrade yet, but this is a library built with the diversity in mind. So in addition to the library, the platform also we use a number of different screening platforms, and also we have a comprehensive set of assays that help us identify and validate that a protein is a direct substrate of the E3 ligase and can be degraded. So as I said, currently, the molecular glue discovery cannot be rationally designed, but as we accumulate more and more data on these structures and the activities, we're accumulating a lot of data, and we're building algorithms to predict what targets can be degraded and how to design these molecules. And hopefully, eventually, we'll be one of the first companies to be able to rationally design molecular glue degraders. So with this platform, we've signed a $1.2 billion research collaboration with Takeda in May for multiple targets. So we're very glad to have this collaboration, and it's a transformative deal for us. With the platform, so how you measure the success of a platform, you have to have programs generated out of the platform that are validated and are worthwhile to advance into clinic. We have a robust pipeline. Our lead compound is the typical undruggable target. It's an RNA binding protein. It doesn't have the pocket for small molecule inhibitors or not even for PROTACs to target, but we can degrade it as a glue using molecular glue degraders. It's been very well published, over 1,000 publications to link it in the cancer metastasis and survival. So we think it has the blockbuster potential. Our second program is a V1 degrader. V1 has inhibitors in clinical development, but inhibitors, kinase inhibitor, can tend to hit multiple kinases, and also the inhibitors in the clinic now also hit PLK1. We think that is causing some of the toxicity. Our degrader binds a different site from the V1 inhibitor, so it avoids all kinases, and we have a very good selectivity and a different mechanism we can see from the data. And we also have GSPT1 degrader as a payload for ADC. So ADC field, they're looking for novel payloads, and degraders is an area that ADC companies are very interested in, because degraders tend to be very potent. It's a catalytic event, and it also can have potentially very good bystander effect for a payload. So these are the more advanced programs. We also have multiple earlier stage programs. I think I'm missing a graph there, but it doesn't matter. So we were founded, we started the company 2021. We raised 2 rounds of capital of $35 million, and we're now raising a Series A+ round of $20 to $30 million. We're a JLabs member and Roche Accelerator member, and we have labs in Shanghai and San Diego, and we've won some awards, including the Aim High Women Entrepreneur Award and Grace Award. So as I said, we have a platform that has generated a robust pipeline. Our lead program is about to enter, we are planning to file IND by the end of this year, in a couple of months, and start clinical studies beginning of next year. And the lead indication we're targeting is BRAF mutant basket tumor and also HCC. We also have WEE1 and GSPT1, as I mentioned, but we also have inflammation programs that are in discovery stage and multiple programs with Takeda. So the Takeda deal, as I mentioned, it's a $1.2 billion deal with upfront R&D funding, research milestones, and development commercial milestones. It involves multiple targets in 3 therapeutic areas: oncology, neuroscience, and inflammation. And also Takeda will make equity investment in the company. So for a startup company like us, this is a very important transformative deal. It not only expands our therapeutic area into from oncology into inflammation and neuroscience, we probably wouldn't do ourselves with our limited resources, but it also, as I mentioned, in the glue field, data is very important for us to understand how to design and advance glues. And with these programs with Takeda, and we also build knowledge and IP for us to advance in the glue platform space. Just a little bit further explanation of the differentiation of our platform. First, I mentioned our glue library. We've designed and synthesized every single library based on this hypothesis that glues, when they bind to E3 ligase, they modify the surface of E3 ligase and create a new surface, and it will recruit and degrade the targets of interest. So with a different chemical structure, we have a different chemical core structure. We have 60 different cores from lenalidomide core. With this, we're in a different chemical space. So this is, I make an analogy to digging mines. We're digging in a different place than most glue companies who use lenalidomide cores. And we're finding different targets because the structure determines what targets we can find. Also, with a differentiated structure gives us a very strong IP position. This I'll skip. The second differentiator is we use multiple approaches to screen for targets. One approach is from compound to target. Especially in the earlier days when we built the library, we start from compound. We do 3 different screenings. We do phenotypic screen. We look for cell killing in cancer cell lines and in CRBN knockout cancer cell lines, and look for the CRBN-dependent cell killing. And then we figure out if a compound can kill a cancer cell. We do proteomics analysis to figure out what is the target. But in proteomics, you can often see downstream effect. So sometimes hundreds of proteins are downregulated, but what is the true direct target? We also do proximity screen. So for glues to degrade, they first need to recruit target to the proximity. And with all the binding data, proximity data, and degradation data, we have a database. This is accumulated proprietary database for us to quickly validate the target and also to advance our SAR. So the second approach we use is from target to compound. Now we have an extensive library. We can use this method. This is the method we use with Takeda. So Takeda gave us a number of disease targets that they are very interested in, but they couldn't target using other approaches. So they want the molecular glue as a toolbox to add into their R&D and to discover drugs for these targets. So we first used our own algorithm to predict which one of those targets are more likely glue substrates. So we selected a few, and we've built, right now it's been a few months, we built a reporter construct, and we conducted high throughput screening. We made very good progress on finding the hits. And we also need to validate, as I said, the hits are direct substrates, and that the function, the mechanism is what we expected. So we are making progress in this area. And the third is the proprietary database and the algorithm. So based on what we know about the glue structures, the target structures from the public space, as well as our proprietary validated glue degrons. Degrons are the part of the structure on the target that are recruited by glues. So we've developed an algorithm for us to predict what proteins can likely be degraded. We've predicted 3,800 proteins are likely substrates of molecular glue. And we actually are realizing now our prediction need to be updated. We're constantly learning new things. I think some of the rules we used initially are no longer true. So we're expanding the algorithm. I think there will be more targets that can be degraded. So the discovery of the first glue hit for a new target is a very high hurdle because the small molecule has to fit just right between the E3 ligase and the target to make it work. So very few companies can discover new glues for a new target. So these are the glue targets in clinical development. IKZF is the first molecular glue target developed by back then Celgene, now BMS, and there are many people doing the derivatives of IKZF. GSPT1, IKZF2, and CK1 alpha and WIZ are currently in clinical now. So from the public data, only 4 companies have discovered glues for new targets, and we're one of the 4 companies, and we have chosen, we have degraders for those 5 targets, but we have chosen to work on some novel targets that we discover ourselves. We think there are better biology and better commercial potential. So HUR and V1 are the 2 examples. So I'll just spend a little bit time on HUR. This is HUR, as I said, it's an RNA binding protein. It is, I make the analogy to MYC. MYC is a transcription regulator. HUR basically regulates the translation of many pro-cancer survival genes. It binds to the 3' UTR of pro-cancer survival genes, mRNA, and stabilizes it. So by degrading HUR, we basically can get rid of a lot of the cancer drivers. So this has been published to be involved in many different types of cancers. And as I mentioned earlier, the HUR protein doesn't have any pocket for small molecule inhibitors or PROTACs to bind. So glue, molecular glue, is a perfect modality to target this very important gene. And this shows some of the data we've generated to show that HUR, we have a molecular glue degrader for HUR. So on the left-hand side, I'll start with graph B. It shows that our glue degrader can reduce the protein level in a dose-dependent manner. But we have to show that it's a direct substrate. So in A, we actually in vitrally reconstituted the ubiquitination. When adding the last lane, when adding the molecular glue, you can generate a polyubiquitin chain. So that's a definitive demonstration of molecular glue mechanism. We also have shown the direct ternary structure formation by showing in C, it's the NMR structure, and on the right-hand side is the cryo-EM structure. And in D, we prove that HUR is the driving factor for cancer cell killing. So the top is the wild-type cancer cell. You can see that it can be killed by the HUR degrader. The bottom half is a non-degradable HUR engineered cell line. So once we make the HUR non-degradable, we can rescue the cell killing. So I'll skip a lot of things. So we did, because this is a first-in-class target, we have to figure out what indication it can play a role. We did bioinformatics, we did a lot of cell line analysis, and of course, animal model also. So we've identified several indications. The most advanced that we're going to advance first is BRAF mutant colorectal cancer. So we've done the analysis in many cell lines and in CRC, in animal models to show the efficacy of this molecular glue in colorectal cancer. We also have seen very good data in hepatocellular cancer. We're working with a hospital. We got 18 tissue, and we established a patient-derived xenograft model from 18 patients. They are HBV-infected hepatocellular cancer patient, and we've seen 8 out of 18 responded to our degrader, similar to lenvatinib, the standard of care, but the patient group responding to our degrader are different from those responded to lenvatinib. So we're currently in the process of trying to find the biomarkers for this patient group. There are also other indications that we have data we can plan to pursue. This just shows the animal model data. On the left-hand side, we show monotherapy, the single agent activity. The top is the CDX, and the bottom is a patient-derived animal model data. So I want to point to the third graph on the bottom right is a large tumor that we can see the very, very obvious regression of the large tumor. So on the right-hand side, we show the synergistic effect with BRAF inhibitor. Right now, the standard of care for BRAF mutant CRC is BRAF inhibitor plus a EGFR antibody. So we show synergistic effect on top of that. I don't even have time. So this is our second program, a V1 molecular glue degrader. V1 is a G2M a checkpoint regulator. So by degrading V1, we allow the cell to go into the cell cycle and accumulate damage. So this will cause mitotic catastrophe. V1, as I said, there are inhibitors in the clinical. So we're comparing our degrader in graph A on the right-hand side. We compare our degrader with inhibitors, and we have better efficacy there. And this shows a V1 as a DNA damage repair agent. It can have synergistic effect with other DNA damage repair agents like ATR inhibitor. Our third program, the GSPT1 molecular glue as ADC payload, Aurum did a deal with BMS with $100 million upfront to have a GSPT1 degrader conjugated to CD33 antibody as they call what we call DAC now, degrader antibody conjugate. So I think this is a really interesting field, and we have very interesting data to show that our degraders are very potent and can be conjugated and have cell killing. So our financing plan, just very briefly, we're raising A+ round of 20 to 30 million now, and we plan to do that by the end of this year. Thank you.
Great, thanks. It was awesome to see the progress since you presented last year. So our final speaker for the company showcase will be Dr. Ronald Herbst. Just continuing the theme of looking at the interface of AI and biotech, in this case looking at a really interesting protein engineering platform. Ronald, prior to joining as the Chief Scientific Officer to Biologic Design, was the head of oncology R&D at MedImmune and AZ for 15 years, and he's trained in both Munich and Stanford. So welcome to the stage, Ronald.
Thank you very much for the invitation, and it's a great pleasure to be here and introduce biologic design in this forum. So as you can see, we are really trying to redefine what antibodies can do as a therapeutic modality using computational methodologies. We heard a lot about AI in the context of translational medicine, target discovery. So here we are using AI and machine learning for the discovery and development of new therapeutic antibodies that have quite unique function. We are not focused on using AI to make antibody development faster or cheaper. It's really all about novel functionality to drive better outcomes for more patients. And here we are really aiming at technology that gives the antibody more than just a single function, that we can do multiple things in a single molecule. And this is really central to our overall approach. All of this started with the founder and CEO of the company, Yanai Ofran. He is a computational biophysicist. When I joined the company, that was the first time I heard about this job description. But he was really one of the pioneers in applying computational technology to the engineering of antibody CDR regions to give them new capabilities. And already over 10 years ago, he founded Biologic Design in Israel, Rehovot, close to Tel Aviv. And just this year, we started the process of actually establishing lab and offices here in the DMV area. We're actually right now operating out of J Labs here in the city, and are looking forward to building up the group and then expanding into a new, more permanent home. The company is also supported by a number of quite talented and capable people. Micha Perlman is our CEO, prior Teva strategy, also worked in the Ministry of Finance in Israel. Tim Wyant, very experienced drug developer, Takeda and others. And then Inbar Amit, who will be joining us for a little bit from Israel here next week. So looking forward to her visit. She's the VP of Drug Discovery, and then very important, the head of our computational team, Zvika Hartman. He is tremendously experienced. He knows the ins and outs. He was actually the founder of Google Health in Israel. So as you can see here on this slide is that we are using AI computational methodologies for antibody discovery and development. And this is actually a clinically validated platform. I will introduce the first molecule that has already advanced to phase 2 in immuno-oncology. And so we are focusing largely on biology that we understand, but think that with our unique capabilities, we can generate molecules that push what has been able to achieve with prior approaches to expand the benefit for patients with severe disease. Obviously, we are very much interested in developing and advancing our own internal pipeline, and I'll introduce a couple of molecules to you in a minute. But we've also been engaged over the years in quite a number of collaborations, strategic partnerships with larger companies such as Nectar, Merck, Lilly, AstraZeneca, and so forth. And just over the past 10 months, we have announced 3 quite large deals. First with Teva, who have taken out and are now partnered a program that we have advanced. Similar Nectar, another antibody program with quite unique functionality. And very recently, this summer, we announced a discovery deal with Merck that utilizes one of our antibody platform technologies for several programs, both in the oncology and autoimmune space. So we are using this technology essentially to advance antibody capabilities in very 2 significant ways. The first modality or approach that we are able to achieve is that rather than generating antibodies against a whole molecule through phage display screening or hybridoma campaigns, our technology actually allows us, if we understand the mode of action, the biology of the target, to already zoom in from the very beginning on the epitope that's really critical for the biology. So these are precision binding approaches against predefined epitopes, and we don't even need target structural information for that. This is quite amazing. So we are not generating antibodies against anywhere else in the molecule. We are only selecting antibodies that from the get-go hit the region that matters the most based on the biology. Sometimes this biology is linked to a particular conformation that is driving agonism or antagonism, or activity or no activity. And so we can also go for particular conformational states and stabilize a molecule in its active or inactive state by design, resulting then in a highly specific, fully human standard IgG with this unique function. The other antibody-based technology that we have developed is to be able to go after 2 different targets with a single standard symmetrical IgG. These are not conventional bispecifics. These are still very standard symmetrical immune globulins, but where each Fab region has the ability with high affinity and specificity to accommodate alternatively 2 different target antigens. This is quite unique. I think we are the only ones who have developed this kind of technology so far. So the antibody can either bind antigen A or antigen B, or a combination of A and B on each arm. That also means that the antibody can adjust to its environment and go after what's needed in a particular environment or state of the disease, or in similar fashion. And I'll give you a couple of examples where this makes a lot of sense, and how we are very excited of pushing these programs forward. And all of this is now reflected also in our pipeline. You see at the top here 3 programs that use this epitope-specific design. The middle 3 are multibody-derived, and then something else that we are in the midst of developing is what we call super engagers. This is also a multibody-derived or multibody-based approach, but now is actually a bispecific, but it's made up of 2 different multibody arms. So now you have 4 specificities in a bivalent antibody. And we'll talk about that later on. I'll quickly introduce the concept that's behind this one. So first up, the program that is most advanced is a program that we actually pursuing through a spin-out company that Biologic has developed a couple of years ago. It's called Aulos. They are located in California. The antibody is now called Aulos, or AU-007. And it's an antibody against a well-known target or a well-known piece of biology, which is interleukin-2. Interleukin-2 obviously is central to immune regulation. Interleukin-2 is also approved as a drug, has to be used at very high concentrations, and thus is also associated with quite significant side effects. So people don't like using it. There has been a lot of effort in harnessing the power of IL-2 through mutational approaches to make IL-2 more specific to the effector cell population rather than the regulatory T cell population. And both of these populations are driven through IL-2 signaling. The difference here, of course, is that T effector cells have a dimeric receptor consisting of the beta and gamma chains. And then the regulatory T cells also have the alpha subunit, so a trimeric receptor that ultimately has also much greater affinity for IL-2. So when we think about treating cancer patients, what we want to do is to drive selectively the effector cells, but not the regulatory T cells. One thing to keep in mind is also that for Tregs, IL-2 is a non-redundant cytokine, so they actually need it. And what Biologic has done is, through the epitope-specific design, through this computational approach, developed an antibody that now binds IL-2 in a very unique fashion, so that the antibody binding completely prevents interaction of IL-2 with a trimeric receptor on the regulatory cells, while fully enabling the interaction of this very small cytokine, still bound with a big antibody, to the dimeric receptor on the effector T cell population. In fact, once the antibody binds, it actually stabilizes the conformation of IL-2 that is fitting with the dimeric beta-gamma receptor composition. Very interestingly, with all the IL-2 muteins that were kind of effector cell biased, trying to reduce the binding to the alpha subunit, what you see in the clinic is that after dosing of these IL-2 muteins, regulatory T cells in the patient's blood still go up. They're not going down, they're going up. In our case, in all of the patients that we've treated, Tregs go down. So that proves, I think, at least at that level, that the biology pans out. We believe that the reason for this phenomenon is a negative feedback loop, because what happens when you stimulate T cells, effector cells with IL-2, they make native endogenous IL-2 that is not mutated, and thus still able to bind the trimeric receptor with very high affinity. And that can create a negative feedback loop. We actually have shown this, at least in preclinical models, and published this. So a very exciting piece of data. And when our antibody binds, totally blocks the interaction with Tregs, fully enabling this, while also at the same time extending the half-life of IL-2 in circulation. Now, this antibody went into the clinic. Here, just one snippet of data. This is a melanoma patient with a rather large lesion in the peritoneum, metastasis, and this is the next scan after the first cycle of treatment, where we see a very nice shrinkage of the target lesion. Obviously, we have more patients. There were 3 arms to the phase 1 study. I cannot show all the data. These are data from Aulos presented at ASCO. And you can see in 2 different scenarios, the antibody was tested as a single agent. And then just to beef up the system early on, there was a loading dose of very low levels of IL-2. And you can see here we have a very nice disease control with several responses here. Not all the responses that have been observed are actually reflected in the waterfall plot as indicated here. There were quite significant responses also observed by PET. And then the other scenario is a slightly different scenario where low-dose IL-2 has been given with every dose of the antibody. So it seems, based on these small numbers, that just a single loading dose is exactly the right way to utilize this compound. It's now in the expansions, including an expansion with checkpoint therapy, of course, and I can't wait to see the data coming out of this. Now, after this example, epitope-specific design, moving on to multibodies, and multibodies make a lot of sense. In every disease setting, almost oncology for sure, but now even in the autoimmune setting, combination therapy is the thing. We need multiple approaches to give the patient the best possible outcome. Also in the autoimmune setting, there are multiple mediators, pathways that are engaged simultaneously. And also the spectrum of this may differ from patient to patient. So to be able to treat more patients more effectively, we need different approaches. And then in the oncology setting, of course, we have resistance mechanisms. We heard this morning about mutations in cell surface antigens that are the targets of therapeutic antibodies, downregulation of molecules, heterogeneous expression, whole areas in a tumor that lack the antigen of interest, things of that sort. So all of this, as you will see later, can potentially be addressed with multibody technology. Just sort of to illustrate the potential of these molecules, just one quick sample. This is not oncology. This is in the inflammatory autoimmune setting. This is an antibody designed to treat patients with Th2-driven diseases. These are inflammatory cascades that are triggered by epithelial damage that then triggers the release of so-called alarmins. TSLP is the most prominent, but not the only one. That then kicks off the Th2 T-cell cascade, ultimately leading to the secretion and production of inflammatory cytokines, most prominent, of course, IL-13. And there are antibodies out there that target each of these 2 things individually approved. But of course, in each case, only a fraction of the patients, maybe 40 to 50% shows a proper response. Many patients don't respond at all or don't benefit sufficiently. So here we have developed an antibody that can target both of these molecules that then would synergize within the pathway. And here's the binding modalities, either TSLP or IL-13, or alternatively, and this is important because, for example, in an atopic dermatitis patient, that patient can have acute lesions where the cascade is just starting to get kicked off with TSLP being prominent, as well as chronic lesions where the whole cascade has already happened, and then IL-13 is there in abundance. The antibody, once administered, will adjust to the various locations and neutralize what needs to be neutralized. And here just one snippet of data. I'll just focus on the right-hand side, looking at in a PBMC assay at TARC is a downstream mediator that can be induced by both TSLP and IL-13. And when you look at the single agent antibodies that are approved, anti-TSLP, anti-IL-13, you will never get complete TARC inhibition. With our antibody, you do. So we are super excited, and this program is now taken into the clinic by Teva. But as I mentioned, there's also ample reason for pursuing multibody technology, going after more than one target in the oncology space for direct tumor targeting, either with an engager, with an ADC, or with an ADCC-enhanced antibody. to overcome antigen heterogeneity, to increase total antigenic density if you have the ability to go after two separate targets that have an overlapping expression pattern, and then also to address resistant mechanisms that are linked to target expression or target mutation. Obviously, there's interest in this space, also in the ADC space. Many companies are already developing so-called bispecific ADCs. Conventional bispecific, as you can see, that's a good idea and it's a good progress, but there are also issues with it, because when we think about the heterogeneous expression of antigens on tumors, tumor cells, in the tumor cell gemisch, if both antigens are present, of course, the conventional bispecific will bind bivalently, full avidity, nice cross-linking and internalization and all of that. But if in this gemisch of tumor cells you have only one of the two antigens expressed, of course you lose the avidity, you lose part of your binding ability, monovalent binding, no cross-linking, and that will also possibly affect internalization of your ADC. In contrast, if you do this with a multibody, regardless of what the antibody encounters in this tumor tissue, cells that have both targets, cells that have only the red one, cells that only have the blue one, in each case, the antibody will bind bivalently, full avidity, cross-linking, internalization, while still being a standard symmetrical IgG, which then also enables better manufacturing and production of the therapeutic as an added benefit. We have already advanced one program, and we are looking here at what we call the BD2 program. This is a multibody that can now bind alternatively either Nectin-4 or Trop-2. I apologize, I think also here in the transition from Mac to PC, we had some hiccups here in the layout. But suffice it to say that obviously Trop-2, we heard about it this morning, both Nectin-4 as well, are clinically validated, approved molecules, and importantly, there's very nice correlation and co-expression of these two antigens in a number of quite important tumor types. Now, you can use this as the basis of an ADC, but you can also use this as the basis of what we call a super engager. Now, a super engager is an actual bispecific in the conventional sense, but it's made up of two arms from two different multibodies. So it's essentially a quadrispecific bivalent antibody. And so one antibody arm would be something like the BD2 molecule that I just introduced. So you can target with one arm both Nectin-4 and Trop-2, or another combination of different tumor antigens, while the blue arm here is actually now the immune engager arm. Now, this is a different multibody that we are working on, where this one arm would be able to either bind and recruit T cells or alternatively NK cells. NK cells are also important when it comes to anti-tumor immune responses. And so here you increase the pool of total effector cells that you can recruit. These two different cell types are also susceptible to different immune suppressive mechanisms. So you can essentially double up in your effector arm, and hopefully together as a whole molecule, this will result in better outcome for patients. And to conclude, I just want to show one snippet of data. Now, this is not yet the full super engager. This is just the BD2 targeting arm, Nectin-4 Trop-2, with a CD3 T cell engager arm. And what you can see here, just to illustrate, we had cells that are only expressing Trop-2 as the target antigen, or only Nectin-4 as the target antigen. We incubated these cells in the presence of human PBMC as a source of effector cells, titrated in our engager with a CD3 arm, and you can see that with very, very good potency, we can kill the Trop cells, but also the Nectin-4 cells. So very much looking forward to advancing this technology to expand our toolbox for generating new therapies for patients with cancer. And with that, I would like to thank you for having me and for your attention.